A surgical robot collects a tumour sample. An automated lab preps it for sequencing. The sequencing machine runs and the data is instantly uploaded to the cloud. An AI agent, armed with complete domain knowledge and standardized pipelines, processes and analyzes it faster than any human could, logging everything in a standardized way. The results are automatically published to a public database, feeding into existing knowledge. AI generates a new hypothesis, instructs the next experiment, and the cycle repeats again and again. And then to top it off, the AI lab assistant does a backflip to flex on the fact it just took your job. Is this the future of scientific research? Is AI taking our jobs? Are we doomed? Well, let's dive in and talk about it. Hey guys, welcome back to the channel and to the second installment in our three-part exploration of AI's transformative role in genomic research and bioinformatics. In part one, we covered the current landscape of AI in the field. These lay the foundations that will shape tomorrow's breakthroughs. If you haven't seen that video yet, I'll add a link in the card as well as in the description so you can check it out. But today in part two, we're fast forwarding into the future, projecting where these technologies might take us over the next decade or two. Then in our final installment, part three, we'll get practical. We'll analyze previous technological evolutions as a roadmap to navigate your way successfully through this AI era. So let's peer into our crystal ball. Where exactly is this AI revolution taking us? The transformation AI has brought about in just the last 3 years defies what most of us thought possible. If I could go back 5 years and talk to my younger self and explain how far things have come, I don't think that younger version of me would believe what I told him. In this video, rather than veer too far into sci-fi speculation, I want to ground this exploration in a more pragmatic approach. What we're going to talk about aren't predictions per se. Instead, I'll call them thought experiments. We're going to follow the logical progression of current technologies, examining persistent problems in the field that demand solutions, and applying reasoning to map out where these trajectories could lead us in genomics research and bioinformatics. So here's the roadmap for today's video. We'll start by unpacking the academic research system, its evolution and current challenges. Next, we'll reimagine science from the ground up, exploring how an integrated ecosystem of AI and robotics could automate everything from laboratory experiments to data analysis and publication, all feeding into a universal, accessible knowledge repository. We'll then explore which scientific domains stand to experience explosive growth in this new paradigm, areas where traditional methods have created bottlenecks that AI-driven approaches could eliminate. And then finally, we'll explore the ethical and societal implications of this sort of transformation and how it could reshape society, influence the balance of power in science, and drive the evolution of ethical standards and regulatory frameworks. So first up, let's talk about the academic research problem. If you've spent any time in this world, then you'll recognize some persistent problems that slow down scientific progress. I see 3 major barriers worth addressing. First, there's gatekeeping. A lot of knowledge is locked behind paywalls. If you're not affiliated with a university or a large institution, getting access to the latest research can be expensive, sometimes even tens of dollars per paper. And even if you do have access, there's another problem: the speed of publication. Peer review can take months, even years, to get a single study published, not to mention the time it takes to write everything up. Sometimes the process drags on simply because of inefficient workflows and outdated administrative practices. Then there's funding politics. Most research requires grants, and getting one isn't just about having a great idea, it's about playing the game. Funding bodies often have very narrow priorities, so researchers have to tailor their proposals to fit those agendas rather than pursuing truly innovative or high-risk ideas. There's also institutional inertia, where big labs with established reputations tend to get funding more easily, while newer, smaller groups struggle to break in. This creates an academic rich-get-richer scenario, where the same ideas keep getting funded, and the truly novel approaches have a hard time getting traction. And thirdly, we have human bottlenecks. Research today still involves a lot of manual work that could be automated. We have some of the brightest minds in the world spending weeks on tasks like writing grant applications, organizing datasets, or cross-checking references, work that takes time away from actual discovery and innovation. The irony in all of this is that science is supposed to be about open knowledge, collaboration, and progress, but the systems we've built around it, whether it's paywall journals, slow bureaucracy, or funding barriers, often make it harder to advance research. So why is this the case? How did this system come to be? And why are we even talking about this? Well, research wasn't always institutionalized. Before the 1800s, research was a luxury. Scientists like Newton and Galileo here depended on wealthy patrons, so kings, merchants, a church, to fund their work. Knowledge traveled slowly through letters and elite societies, with no systematic approach beyond individual curiosity. The turning point came in 19th century Germany, with Wilhelm von Humboldt's revolutionary university model, combining teaching and research. This approach created powerful advantages. Students learned directly from active researchers, and knowledge production became systematic rather than dependent on isolated brilliance. The model proved so successful that it spread globally. This institutionalization brought critical structure to science. Peer review emerged as a quality control mechanism, a way to validate findings before they entered the scientific record. While today we see its limitations, it remains our primary defense against fraudulent or flawed research. The post-World War II research boom further formalized the process. Government funding created unprecedented opportunities for discovery while establishing necessary accountability measures. These structures, grant proposals, ethics reviews, and progress reports help maintain standards and ensure research serves public interests rather than just satisfying individual curiosity. The challenge we face isn't that these systems exist, they're essential for scientific integrity, but that they were designed for an older era. The rapid expansion of the internet, the technology boom, and now the AI revolution have dramatically accelerated the pace of discovery. Yet our research systems remain grounded in frameworks developed for a slower, analog world. We've built a research infrastructure optimized for careful, deliberate progress, qualities indispensable to scientific rigor. However, as computational power and data generation scale exponentially, these human-centric systems struggle to adapt. AI offers a chance to bridge this gap, not by dismantling the structures that uphold scientific reliability, but reimagining them for the modern era. It has the potential to enhance efficiency while preserving the core principles that ensure research remains reproducible, ethical, and trustworthy. Let me paint a picture of what this might look like in the not too distant future. Remember our opening scene? A surgical robot collects a sample, an automated lab preps it and sequences it. The data uploads instantly to the cloud, where an AI agent processes, analyzes, and publishes the results to a public database. This isn't a sci-fi fantasy. Many of these components already exist in isolation. High-throughput labs already utilize liquid handling robots that can prep hundreds of samples with precision exceeding human capabilities. Standardized computational pipelines already exist for many common analyses. What's missing is integration, connecting each part of the pipeline into a seamless, intelligent system that can take a biological sample from wet lab all the way to public knowledge with minimal human friction. And it's this integration that AI may in the future be able to bridge. Imagine a world where you design an experiment in the morning, and by the time you wake up the next day, not only has it been run, but the results have been analyzed, contextualized against existing literature, and published to an open access repository. That's not just faster science, it's science at a scale that we've never seen before. But I'm sure you have lots of questions. So let's break down this pipeline further, starting with this part on publication. Right now, publishing scientific research is an epic journey. From experiment to final paper, it can take anywhere from several months to several years. Writing, formatting, submission, peer review, revision, resubmission, all to achieve what? The accurate sharing of robust and validated scientific findings with the community at large. I think with AI, we can achieve this same end goal, but in a fraction of the time and with more accuracy and confidence. Let's break down how. So let's introduce AI agents. AI agents can be thought of as autonomous systems that perform tasks without constant human supervision. They can take complex goals and break them down into steps, making decisions based on data and adjusting their approach accordingly. Today we have early agents capable of writing code, analyzing data, and even coordinating tasks across platforms. Fast forward 5 or 10 or 20 years, and you're looking at systems that can interpret raw experiment data, run quality control checks, compare results against existing literature, flag anomalies, generate comprehensive human-readable reports, suggest follow-up experiments or missing metadata, and then package everything for upload into an open access repository. And they can do it instantly. Now, I know what you might be thinking, but how do we trust the results? How do we validate experimental design? What about fraud, novelty, the significance of findings? And this is where the real innovation happens. AI won't just publish the data, it would review it too. Imagine multiple AI models or agents trained differently that could review the same dataset, flag inconsistencies, and then run statistical robustness checks in parallel. AI can then instantly compare your results against every other published or unpublished dataset in the field, identifying possible contradictions, overlaps, or breakthroughs. Agents could tailor explanations of results to different expertise levels, beginner to expert, on what the results mean and how confident we should be in their implications. And just like GitHub, all results would be tracked in version controlled databases. If a dataset is challenged later, its lineage and edits are traceable. Think scientific commit history. This doesn't mean humans are removed from the loop, it means they're moved up the loop. Instead of manually proofreading figures and formatting citations, human experts would focus on strategic oversight, reviewing the reviewers, validating the validators. AI-assisted peer review wouldn't eliminate rigor, it would amplify it. And here's a fun thought. We don't even need to stick to the old idea of a paper or journal. Why write 10 pages of static text when an AI agent could generate dynamic, interactive dashboards for every study? Imagine portals where you can drill into the raw data, rerun the AI analysis with different parameters, view confidence scores for every result, ask natural language questions about the study, and get citations, background context, and suggested readings all generated on the fly. In this world, knowledge isn't locked in PDFs. It's a living system, searchable, testable, remixable. All of this leads to one simple but radical idea. What if the time from experiment to publication wasn't years, but hours? What if anyone in the world, from a top tier lab to a high school student with an internet connection, could access and build on the latest genomic discoveries the same week they were made? That's not just faster science, that's science at scale. And yes, it sounds wild, but given where agentic AI is headed and what's already possible with today's tech, I don't think this is too absurd if you look long enough into the future. Let's zoom out for a second. If science becomes something that updates like open source code, versioned, forkable, reproducible, then we have to rethink the entire research pipeline. Not just how we publish results, but how we generate them. From lab bench to data analysis to public access. So here's the vision. A fully automated pipeline where wet lab experiments, dry lab computation, AI-led publication, and knowledge integration are stitched together into one continuous loop. A system that feeds itself. It all starts in the automated wet lab, robotic platforms that can pipette, mix, incubate, and sequence with precision. These systems can run thousands of experiments in parallel, track every variable down to the microsecond, and never get tired or make typos in the lab notebook. But these aren't just dumb machines executing scripts. They're connected to AI-driven experiment designers, agents that analyze the current state of the knowledge graph, ask, what's missing, what's unclear, where's the next most valuable place to probe? These agents propose hypotheses, design protocols, and then send them to the wet lab for execution. As experiments run, real-time data flows into the dry lab pipeline. There, AI models clean, process, and analyze the data. Multimodal models, trained on everything from DNA sequences to cell images to scientific papers, interpret the results in context. If something looks off, the system flags it, suggests troubleshooting steps, or even reroutes the pipeline to run a variation of the experiment. Once validated, the findings are packed into a machine-readable publication. Think of it as a commit to the Universal Knowledge Graph. Not just a summary PDF, but the full suite. Raw data, analysis code, metadata, methods, every reagent, every parameter, every step, logged and timestamped. No more, we incubated for 2 hours. It would be incubation began at 2:32 at precisely 37.2 degrees Celsius and blah blah blah, but in a structured format. You could replay it like a simulation or fork it like an open source project. Today we face a crisis where around 50% of studies can't be replicated. The reasons vary, either human error in recording methods, missing metadata that would be crucial for reproduction, and what I'd call lazy workflows where corners are cut to save time. In our hypothetical integrated system, it would transform the reproducibility crisis into a reproducibility revolution. The bottleneck wouldn't be information sharing or methodology documentation, it would simply be the time it takes to run the experiment itself. And because it's all structured and standardized, the knowledge graph updates itself. It's not just storing information, it's reasoning over it. The graph highlights gaps, AI agents design new experiments, and the loop spins forward, a closed feedback loop of discovery. This is more than just reproducibility, it's scientific compounding, a system where every experiment makes all future experiments smarter. Okay, that's enough sci-fi pondering for now. Let's go on to which scientific frontiers could see explosive growth. One of the most exciting ideas is this idea of digital twins, a virtual model of an individual's biology built using data from their genome, transcriptome, proteome, metabolome, microbiome, and more. The concept originated in aerospace and manufacturing to model jet engines and industrial systems. But in bioinformatics, it takes on a profound new dimension, a fully interactive, real-time simulation of a person's body. Or imagine a digital twin of a cancer patient's tumour, for instance, which could incorporate their unique genome mutations, the tumour microenvironment conditions, epigenetic states, drug sensitivity, historical treatments, and more. But this isn't just a passive database or 3D model. With enough biological data and advanced AI modelling power, it becomes an interactive simulation environment, a conversation partner. A clinician might ask, what if we apply a combination of drug A and a low dose of immunotherapy B and instantly receive a prediction of the likely outcomes based on the patient's unique biology, integrated knowledge of signaling pathways, and similar cases across a global dataset. This enables in silico experimentation at unprecedented scale. Treatment plans could be trialed virtually before ever reaching the patient, reducing risk and improving efficacy. Novel drug combinations, some that might never have been considered in traditional workflows, could emerge through AI-driven screening across thousands or even millions of digital twins simultaneously. And as AI continues to improve its ability to model biological systems with high fidelity, this opens the door to hyper-personalized medicine, where we no longer rely on population averages, but instead tailor interventions to the exact molecular and phenotypic state of you, not just someone like you. Now, of course, building such digital twins is no small task. Biological systems are extraordinarily complex, with interactions spanning from the genome to the proteome to the environment and behavioral factors. Simulating this dynamic, multi-scale system requires massive computing resources and sophisticated AI architectures capable of reasoning across time, space, and modality. Although we haven't reached this point yet, progress is being made. Breakthroughs such as GPU acceleration, high-dimensional data integration, and quantum computing may turn these visions into a reality within our lifetimes. But to build such rich, dynamic models of biology, we need more than just raw computing power. We need AI that can actually understand biology in all its messy, multi-dimensional complexity. This is where a new breakthrough in AI comes in: multimodal contrastive pre-training. At its core, this method is a way of training AI systems to understand multiple types of biological data at once, and more importantly, how those data types relate to one another. Most traditional AI models are narrow. They specialize in just one thing: analyzing DNA sequences, interpreting medical images, or parsing scientific papers. But multimodal contrastive pre-training takes a broader approach. It learns by drawing connections across modalities. For instance, it might learn to associate a microscopy image of a cancer cell with the gene expression profile that created it, or connect a protein's 3D structure with how it's described in scientific literature. This matters because biology is inherently multimodal. Genes, proteins, cells, tissues, and clinical outcomes are all interconnected, operating within dynamic systems that span many layers of information. Models trained with multimodal approaches can bridge these layers, enabling AI to generate insights that would be impossible to find by analyzing just one data type in isolation. This kind of cross-modal reasoning is foundational for building digital twins. To simulate real biology, AI must synthesize vast and diverse datasets. Multimodal contrastive pre-training provides the scaffolding for this unified understanding, allowing AI to learn more like we do, by connecting the dots across many types of input. And unlike traditional rule-based systems, these models improve as more data becomes available. Every new modality, dataset, or paper strengthens the web of associations that AI can use, pushing us closer to truly intelligent biological systems. And while these capabilities aren't a reality just yet, this is definitely an area that will attract increasing attention over the next couple of decades. If we're able to achieve these feats, the impact could be enormous, unlocking breakthroughs in personalized medicine, accelerating drug discovery, and deepening our fundamental understanding of life itself. Okay, let's explore some more ideas, this time in more specific domains. Firstly, mapping the brain at scale. The human brain has about 86 billion neurons and nearly a quadrillion synapses. Mapping this entire network, what scientists call the connectome, has long been one of the biggest challenges in science. It's like trying to chart every road, alley, and traffic pattern in a city of galaxies. But AI is making real progress. It can now analyze terabytes of 3D brain scans, classify individual neurons, trace their connections, and begin to reconstruct functional circuits. With models trained on these massive brain maps, we could eventually simulate neurological disorders, decode aspects of consciousness, or even develop personalized brain-computer interfaces. Again, this one feels very far away, but interesting to think about nonetheless. Okay, the next idea pertains to synthetic biology. In synthetic biology, researchers engineer living systems, whether it's programming bacteria to produce insulin or redesigning yeast to make biofuels. But traditionally, this work has involved a lot of guesswork, trial and error, and deep domain expertise. AI could improve this. Advanced models trained on genomic and metabolic data could simulate, predict, and suggest biological designs. For example, instead of manually tweaking genes, you could ask AI to design a genome that produces molecule X with high yield and stability. The AI could generate a blueprint optimized for stability, expression, and metabolic performance. And then robotic DNA assembly systems could build and test it in real time, cutting development cycles from months to just days. This approach mirrors software development, moving toward compiler-level biology, where high-level goals are translated into genetic code with AI managing the complexity. Now, another idea is Predictive ecosystem modeling. Ecological systems are notoriously non-linear and chaotic, but with AI advancements and multimodal integration, AI could integrate satellite imagery, weather data, biodiversity records, and a network of environmental monitors to predict changes in ecosystems even before they happen. Imagine forecasting a coral reef bleaching weeks ahead or spotting early signs of species collapse. This could turn conservation into a proactive rather than reactive effort. Okay, one more idea now. Imagine a handheld device, something small and sleek, maybe even used by your doctor. You press your finger on it, and in seconds, it sequences your DNA, sends it securely to the cloud, and runs it through advanced AI models. It cross-references your genome, health history, and lifestyle data. Within minutes, it returns tailored insights, or maybe even recommends treatment designed just for you. Of course, we've heard bold predictions like this before. Some of them, as in the case of Elizabeth Holmes and Theranos, didn't quite pan out. But the difference now is that the science is actually catching up. Technologies like nanopore sequencing already make portable DNA sequencing possible, and with AI-powered cloud-based analysis, we can interpret complex biological data faster than ever. With further improvements in speed and usability, this kind of personalized, on-the-spot health insight could become a reality sooner than we think. Okay, it's all well and good to paint this sci-fi picture of robots running experiments, AI agents publishing results, and advanced biological simulations, but let's get real for a moment. Between our current research landscape and this automated future lies a very real and messy tangle of ethics, politics, and practical limitations. Let's start with a big one: power. Advanced AI chips, robotic labs, high-throughput sequencing, they're not cheap. Right now, access to these technologies is mostly limited to well-funded universities, big pharma, and tech giants. That creates a paradox. AI might democratize data analysis, but the physical infrastructure to do cutting-edge research might stay locked behind elite institutions. So how do we ensure fairness? One answer might be global research consortiums, so shared cloud labs or AI resources accessible to underfunded teams. Or new international organizations that fund and maintain open research infrastructure, kind of like the World Health Organization, but for AI-driven science. Because if we're building the future of science, it should be accessible to everyone. But access isn't the only issue. There's also the question of what gets automated and what doesn't. So not everything will be taken over by machines, at least not in the short term. Field work, clinical sampling, complex ethical decisions, those are messy, unpredictable, and very human. And while AI is great at spotting patterns in existing data, the generation of truly transformative hypotheses that open entirely new research directions may remain human-driven for the foreseeable future. So a more realistic progression would be a complementary relationship rather than a competitive one. Humans providing creative direction, ethical oversight, and novel hypotheses, with AI handling data processing, pattern recognition, and routine experimental execution. And this brings us on to another question. Who should get credited for research if AI runs an entire experiment on its own? Maybe it's a shared system. You could have fully autonomous, routine experiments running 24/7, while human researchers lead the design of novel studies upstream. In either case, we'll need transparent systems to credit both computational and human contributions fairly. And speaking of systems, how might the current funding of research change? Today's grants are slow, bureaucratic, and built for research timelines measured in years. But if AI can compress experiments into weeks or even days, the funding model has to evolve. Blockchain technology offers interesting possibilities here for creating transparent, tamper-proof records of where funding goes, how it's used, and what it produces. And as a bonus, would lead to less paperwork, more accountability, and fewer biases. AI might also help to make the grant proposal process more strategic. For example, if multiple teams are proposing similar ideas, an intelligent system could suggest shared infrastructure or collaboration to avoid duplication and make better use of resources. But of course, there's always a balance to strike when it comes to sharing information. Researchers already have concerns about being scooped or losing a competitive edge, so while AI can facilitate collaboration, the system would need to be designed carefully with protections in place to avoid putting ideas at risk before they're ready. But that's all still speculative. What's not speculative is the significant privacy challenges in genomics. Unlike a password, you can't change your genome if it's compromised. And unlike conventional medical data, your DNA reveals information about your relatives, past, present, and future. These characteristics mean traditional privacy and consent models fall short. And yet, if we want to build a truly AI-driven research ecosystem, then genomic data has to be a part of it. But how we handle it matters. That means designing systems with privacy and security built in from day one. Traditional anonymization techniques often fail to meet the challenge because because genomic data is inherently unique to individuals. Even de-identified genomic data can sometimes be re-identified when combined with other publicly available information. In other cases, especially for highly sensitive or regulated data, contributing directly to a central system won't be possible. Private companies invest heavily in research, and they have valid reasons to keep certain insights proprietary. But that doesn't mean those datasets are excluded from progress. This is where tools like federated learning come in. Instead of sharing raw data, institutions can train AI models locally on their own servers, and then they share only the learned parameters, not the underlying data, so they can contribute to the broader scientific effort without compromising privacy. Differential privacy is another solution. This introduces a small amount of statistical noise to data or model outputs, keeping overall trends accurate while protecting individuals from being identified. It's especially useful when even aggregate insights could be sensitive. So the future may not be one universal database, but a network of systems, some open, some restricted, connected by shared standards and privacy-preserving AI techniques. Imagine something similar to REST APIs for genomics, standard interfaces where any sequencing data, regardless of its source, adheres to consistent formats and access patterns. This way, even private or siloed data can contribute to global discovery. In fact, there's been growing excitement around new technologies like Model Context Protocol, or MCP, which aims to make it much easier for AI models to interface with diverse data sources. By providing a standardized way for models to securely access, interpret, and learn from data across different platforms, MCP would help break down silos and accelerate collaborative research, while still respecting privacy and data ownership. But privacy isn't the only hurdle. We also have to consider bias. Right now, many genomic databases overrepresent people from European ancestry. If we're not careful, precision medicine could end up working great for some populations and not at all for others. Addressing this requires both technical innovation, like improving predictions from limited or imbalanced datasets, and policy intervention, so incentivizing diverse data collection and requiring fairness evaluations for AI-driven health tools. And then there's the dual-use dilemma, the idea that the same AI systems that help us cure disease could also be used to design biological threats. That's not hypothetical. It's a genuine concern that requires serious governance. We need clear rules, intentional cooperation, and ongoing oversight to ensure we're steering this tech towards progress, not peril. That said, change is hard, especially in academia. If you spent years climbing the ranks, mastering the system, and finally getting to a place where you benefit from it, you're probably not going to be the first in line to challenge the status quo, and that's understandable. Some researchers might think, if it ain't broke, why fix it? And that's a fair question. But if AI can cut through bureaucracy, eliminate redundancy, and speed up discoveries in science, where breakthroughs can save lives, then maybe the real question is, how long can we afford to wait? Given all of these challenges, I think a more realistic vision involves the gradual integration of AI throughout the research process, automating routine tasks, enhancing human capabilities, and streamlining communication. Rather than a sudden revolution, we're more likely to see an accelerating evolution towards a more efficient system, one that hopefully preserves scientific rigor while removing unnecessary barriers to progress. So that brings us to the end of this second installment. In this video, we've explored a bold vision of the future, where AI and automation reshape genomic science from the ground up. Think robotic labs running experiments, AI agents publishing research in real time, and breakthroughs in multimodal biology and digital twins. We've also unpacked the bottlenecks of today's system. That includes funding politics, gatekeeping, inefficiency, and how AI might help us overcome them. But with all this change, one question still remains. Are robots and AI agents really coming for our jobs? The reality is a lot more nuanced than a simple yes or no. Some roles will be transformed or even phased out. The technician pipetting all day, or the analyst running the same bioinformatics pipeline over and over, those kinds of repetitive tasks are prime for automation. It's also the subject of our next video, where we'll look at how past technological revolutions reshaped the way we work, not just in science but across every industry, and what lessons we can take into this new AI era. So if you're curious, concerned, or just figuring out how to stay relevant, don't miss it. Until then, I'd love to hear your thoughts. Do you see AI as a threat or as an opportunity for scientific research? What aspects of this future vision excite you or concern you the most? Let me know in the comments below, and I'll see you in part 3.